Today we're going to talk about section 1.3, the methods of science. The main idea of this section is that biologists use specific methods when conducting research. Some people call it, as a matter of fact, most people call it, the scientific method. Okay, the first part of the scientific method is something that we all do every single day. We observe the world. Scientists are often very curious about why things happen, how they happen, what makes things happen, all of that stuff. And when scientists observe the world, they usually come up with some question or some problem that needs to be solved. When they do this, they make a hypothesis. It's a possible solution to the problem or a possible answer to the question. So possible explanation based on observations and prior knowledge. Probably a lot of you have heard the word hypothesis before, and oftentimes when you're knee-high to a grasshopper, they will say, what's a hypothesis? It's an educated guess. Well, what does that really mean? What it means is you take all the stuff that you've learned up until that point, and even stuff that other people have learned, and you use that to come up with a possible explanation. So it's all based on prior knowledge. If you go into a room and you flip the light on and the light doesn't come on, sometime in your past you probably realized that maybe it wasn't the power was out, maybe it's just the light bulb was burned out. That's using prior knowledge to solve a problem. That's making a hypothesis. So what do? You go and get a light bulb and you change it. And if it works, great. If it doesn't, it doesn't. That's an example of using scientific method. So you've used scientific method like a lot in your life. You just didn't call it that. That's all. So hypothesis is a possible explanation based on observations and prior knowledge. The thing about a hypothesis, for it to be scientific, it has to be testable. If there's no test for it, then it's not a scientific hypothesis. For example, if you mix red and blue paint, you'll get purple. Well, okay, that's absolutely testable. You get yourself some red paint, you get yourself some blue paint, and you mix them together, and if you get purple paint, that's great. If you don't get purple paint, that doesn't mean that it's not a hypothesis. It just means your hypothesis was wrong. That's all. Another example would be the Boston Bruins are the best hockey team ever. Hypothesis? No, just my opinion. It's not necessarily testable. It's really just an opinion. It's not even a scientific fact. So you have to be a little bit careful about when you're making hypotheses. So the Boston Bruins are the best hockey team, although might be a true statement, might not be. It's not a hypothesis. So the reason that it's not is because it's not testable. Okay, the third part of scientific method is designing an experiment to test your hypothesis. So when I told you it had to be testable, this is where you're going to do that. So an experiment is just a set of repeatable steps that is used to test that hypothesis. And there are some certain parts to an experiment. If it's scientific, it has to have these parts. So the common parts of a scientific experiment are you have to have a control group. That's a set of experimental subjects, and they have exactly the same conditions as all the other groups, and they are used to compare the experimental group results to. So control group is really, really important. If you didn't have a control group and you didn't have something to compare the experimental group to, for example, if you wanted to find out if plants grew better with Miracle-Gro, which is a fertilizer that you can put on them, if they grow taller with Miracle-Gro, you could do a little experiment. If you gave every single plant Miracle-Gro, how would you know if it actually helped? It might not have. So you need something to compare that experimental group to, and that would be your control group. If you don't have a control group, as I said before, you can't really tell if there's a change or not, so your experiment wouldn't be valid. The experimental group is a set of experimental subjects that have one condition different from the control group. So if you were doing a scientific experiment to see if bean plants grew taller with Miracle-Gro than without, You would have some that you give Miracle-Gro to and some that you do not. The ones that you give Miracle-Gro to, you'll record the data, and then you're going to compare that data to the data you've gathered from the control group. So the experimental group and the control group have exactly the same conditions. If we're doing the bean experiment, you would have the same type of beans, the same number of beans, the same amount of sunlight, the same amount of soil, the same amount of watering. It's just one of the experimental group would just get Miracle-Gro rather than just straight water. Another piece of an experiment is an independent variable. That one single condition that the experimental group has, but not the control group, that's called the independent variable. You decide what the independent variable is. It can only be one in each experiment, because if you have more than one, for example, if we were looking at the beans again, and we gave the experimental group not only extra Miracle-Gro, not only some Miracle-Gro, but also extra sunlight at the same time, and you got these giant tall plants, you wouldn't really know which one actually caused that. It might have been the extra sunlight, it might have been the Miracle-Gro. So you can only have one independent variable at a time in an experiment. Going along with the independent variable, experiments have a dependent variable. Dependent variable is just a fancy word for data. These are the results from the independent variable, whatever the independent variable caused. So it's also called the data, aka data. Speaking of data, the next part of the scientific method is you need to collect that data. Data is just information gained from an experiment. You decide what it's going to be. If we were talking about the change in height of plants, the measurement of the heights would be the data. Sometimes it's not something that you can put a number on, like a measurement 55 centimeters. Sometimes it's just a description of what's happening, like it turned from red to blue. So there are two types of data. The first type is quantitative, quantities. These are measurements and numbers, heights, weights, number of beans, number of offspring. All of these different things are quantitative. Next year when you take physics class, you'll have a lot of experience with quantitative data, because you'll be doing a lot of that in physics. We do that some in biology, but oftentimes in biology we have what we call qualitative data. Those are just observations. If you can't collect quantitative data, like a lot of things are difficult to put a number on, then you have to rely on qualitative data, and that is why it's really important that you're very, very good with your observation skills. Once you have that data, then you have to analyze it. Well, what does it mean? Does it support the hypothesis? Does it not? And then you draw a conclusion. In the conclusion, you have to revisit the hypothesis. Yes, the data does support the hypothesis. The beans were taller when they were given Miracle-Gro than not. So that's if it doesn't, that's okay too, because a lot of science is built off hypotheses being wrong. Just because you make a hypothesis, you didn't know what the result was going to be. Why would you need a hypothesis if you already knew the result? So if it's wrong, that's okay, because you can revise your hypothesis and begin again. Scientists have this really great kind of cycle going. Once they get to the end, get to the conclusion, then all kinds of new questions pop up. Maybe their hypothesis is wrong. They have to go back, change it, retest, reanalyze, conclude again, and so forth and so on. Big, huge circle. So I guess that's it for now. Thanks for listening.